Today our lesson is about transcription. The key concept of our lesson is transcription converts a gene into a single-stranded RNA molecule. Now, here we will learn how the RNA carries DNA's instructions. Okay? After the DNA is replicated, the RNA will carry these instructions. Let's learn how this will happen. First, I would like to mention that Francis Crick defined the central dogma of molecular biology, and central dogma is mainly a principle. This principle states that information flows in one direction from DNA to RNA to proteins. Actually, I'm talking about the protein synthesis, okay? So, during the protein synthesis in the cell, the information will flow in this way, only in this direction, from the DNA to RNA to proteins to make the proteins. Alright, as you see here, we have the three processes, DNA to RNA to protein. And of course, the DNA is located in the nucleus. So, the three processes of the central dogma are replication of DNA, which happens in the nucleus of the eukaryotic cells, transcription of RNA, which means converting the sequence of a DNA strand into an RNA messenger, okay? So, transcription also happens in the nucleus of the eukaryotic cells, and after this transcribed RNA formed, it will continue to make the protein by translation, and this RNA molecule will go out of the nucleus to the cytoplasm. So, replication and transcription happens in the nucleus of the eukaryotic cells, while translation happens in the cytoplasm of the eukaryotic cell. Whereas prokaryotic cells have no nucleus, and as a result, these three processes happen in the cytoplasm. So, the main point here you have to keep in mind that RNA is a link between DNA and protein. So, it is an intermediate molecule. RNA is intermediate molecule between DNA and RNA. Okay? Between DNA and proteins. Because DNA only gives instructions, okay? And RNA is the messenger to make these proteins. Now, how these both molecules differ? There are three major ways. RNA has ribose sugar instead of deoxyribose sugar. RNA has uracil instead of thymine, which is the nitrogen-containing base. And RNA is a single-stranded structure, where DNA is a double-stranded helix structure. Now, if you look here, this is the ribose sugar. This one, it has extra oxygen atom, and this is the uracil instead of thymine. If we look at the nucleotides of DNA and RNA, we will see the difference here. In the DNA, we have the deoxyribose sugar, phosphate, and the base. Bases in the DNA are adenine, guanine, cytosine, and thymine. While in the RNA, phosphate group also is there, and ribose, not deoxyribose, ribose, okay, sugar, plus the base, but the bases here are different in the uracil. Instead of thymine, we have uracil in the RNA. Now, as you see here, the DNA is double-stranded, while the RNA is single-stranded molecule. And here are the bases in each one. Now, transcription makes three types of RNA, okay? The first one, transcription copies DNA to make a strand of RNA. Let's see now what are the three Now we will learn about the three types of RNA after we talk about the transcription process. Now, how the transcription process happened? As we learned in replication, the DNA polymerase enzyme is needed to build the DNA, to make a new DNA strand. Okay? Similarly, the transcription also is catalyzed by an enzyme especially to build the RNA, and it's called RNA polymerase. Now, this RNA polymerase and other proteins form a transcription complex. Okay? This transcription complex recognizes the start of a gene and unwinds a segment of it. This complex should recognize a start point of a gene and an end point of a gene. It doesn't copy all the DNA as in replication. It just make copy or transcribe a part of it, a segment on the DNA, a gene. As you see here in this figure, here is the starting site. The RNA polymerase come to this site, recognize the starting site, and this happens according to the demand of the cell, okay, the need of the cell. The cell needs certain protein, so this polymerase go to certain site on a gene, on one strand of DNA to copy it, okay? And here, as you see, there are free-floating RNA nucleotides, which are guanine, cytosine, adenine, and uracil. Okay, here is the complex. Can you see the complex? It's very big molecule, this one, the beige color. All right, now nucleotides pair with one strand of the DNA, only one strand to form the RNA. So RNA polymerase bonds the nucleotides together, as you see here. Here are the floating nucleotides, and the RNA polymerase start to bond them together on one strand of DNA. Okay, the DNA helix wins again as the gene is transcribed. Okay, now the RNA strand detaches from the DNA strand once the gene is transcribed, as you see, and it will start to leave the nucleus. All this happen inside the nucleus. This will start to leave the nucleus to go out to the cytoplasm. Now, what are the three types of RNA that are transcribed? We have three types. The first one is the messenger RNA, and we call it mRNA. It carries the message that will be translated to form a protein. The second type of RNA is a ribosomal RNA, and as we learned, the ribosomes are structures in the in the cell that are factories for proteins. So there is a type of RNA called ribosomal RNA, and they are part from the ribosomes. They help in protein synthesis. The last one is the transfer RNA, tRNA. This tRNA brings the amino acids from the cytoplasm to a ribosome. Why they bring the amino acids? To build up proteins, because proteins are polymers of amino acids monomers. Now, transcription process and replication process, how they are similar and how they are different. Let's see how they are similar first. Transcription and replication both involve complex enzymes. قلنا بالreplication they involve DNA polymerase, وبالtranscription they involve the RNA polymerase. They are complex enzymes in the help of other proteins, and also they involve complementary base pairing. Adenine, guanine, cytosine, thymine in the replication on the DNA, while on the RNA they use adenine, uracil, guanine, and cytosine. Okay. Also, they involve unwinding of the double helix in both processes. Both processes also occur in the nucleus of eukaryotic cells. Now, as you see here, okay, replication, we said that it happens once during the cell cycle of the cell. all right? Because the cell needs only to make one copy of the DNA to be divided, all right? While the RNA can be transcribed several times because it is transcribed according to the demand of the cell. If the cell needs certain protein, it will keep copying and making transcription till it covers all the need of the cell. So RNA can happen at the same gene at the same times, okay? For many times it can be transcribed according to the need of the cell, of this protein copying. The two processes have different end results. How they are different? Now, replication copies all the DNA. Why? Which ensures that each new cell will have one complete set of genetic instructions, while transcription copies a gene, which means a segment on the DNA. Now, replication makes one copy only. Replication occurs only once during each round of the cell cycle because each cell needs to make only one copy of the DNA. While transcription can make many copies of gene because a cell may need hundreds or thousands of copies of certain proteins or the ribosomal RNA and transfer RNA molecules needed to make proteins. Now here are the questions. How are the processes of transcription and replication similar? Both occur within the nucleus of eukaryotic cells, are catalyzed by complex enzymes, involve unwinding of the double helix, involve complementary base pairing to the DNA strand, and are highly regulated by the cell. This is a very important point you have to know, that during replication and during transcription, there will be checkpoint to make sure that no errors happen, okay? So it is highly regulated by the cell, both processes. Thank you for listening and see you next session.